This is a little recap of all of the chapters that we talked about for exam 1. So starting off with chapter 1 kind of read like a little history lesson of microbiology. We talked a lot about the different scientists. Here you can see that I listed the scientists and their contribution. Again, you don't need to know a lot about their life history, just a little bit about their contributions. So if you like flashcards, it might be a good idea to make up some flashcards with the name and their contribution on the other side. So you will need to be able to recognize their name and contribution. In this chapter, we also talked about some different definitions. One of the things that we talked about was aseptic technique. We also had the opportunity to practice that in lab as well. So remember, aseptic technique, this brings in 3 parts. So it's not contaminating yourself when you're working with a culture, not contaminating your surroundings, or contaminating your culture. I mentioned that if you work in healthcare, you're already doing aseptic technique when you're working with a patient. So you don't want your patient to contaminate you. You don't want to give your patient something else, and you don't want to contaminate the surroundings. The reason we used aseptic technique was that we wanted to obtain and maintain a pure culture. A pure culture is going to be free from contamination and only contain one type of organism. We also had the opportunity to observe some colonies in our Ubiquity lab. So if you look at the picture below, you can see there are different organisms. Those different colonies tell us how many different bacteria we've actually captured. So each of those little islands, as I call them, are colonies. And that was one bacteria that continued to divide. A couple of other definitions we talked about. We talked about chemotherapy, which is really a generic definition, and that's really any chemical substance used to treat a disease. Specifically, we're going to be talking about chemotherapy that treats bacterial diseases or diseases caused by microbes. So we'll talk about antimicrobial therapy that we commonly think of as antibiotics. So that is going to be to treat diseases caused by bacteria. So in chapter 1, there's quite a few definitions, also the scientists. So again, if you're a flashcard person, you may want to make up some flashcards or just some different ways to recognize the names and the vocabulary. Chapter 4 was about microscopy. The organisms that we're looking at this semester are very small. They're prokaryotic, very simple cells. And so in order to see them, we need to use our compound light microscopes. So we first started off with this chapter talking about the difference between magnification and resolution. Magnification makes things bigger. Resolution makes things clearer so that we can see 2 cells sitting side by side and see them as separate units. One of the things that we practice with when using our microscope was adding oil. So a lot of times if we've used the microscope before, we might not have gone up to that oil immersion lens because as we go up in magnification, the refraction increases, so the light bends. We need to correct for that refraction, and that's why we add that oil. And it really does improve that resolution so that we can see that clarity. Now, we spent a lot of time working with our microscopes. They are compound light microscopes that have 2 lenses. That's why they're called compound microscopes. So we can see our very small bacteria, but if we wanted to view a virus, we would need a very powerful microscope, an electron microscope. We would not be able to view them with our compound light microscopes. Another thing that we talked about was preparing slides and staining. So if you ever looked at pond water underneath the microscope, you were making a wet mount slide. So you could actually see those little pond swimmy things moving around. We are not going to be able to see our bacteria moving. So we are going to make smears and then heat fix them. Now, the reason that we heat fix them to the slide is that A, it kills the organism. B, it's going to allow them to stick to the slide because we're going to be doing some rinsing and applying of stains. And the other thing is that the heat actually opens up that cell wall. That allows that dye to actually penetrate into that cell wall. So those are the 3 reasons why once we make our smears, we actually have to heat fix them. Now we're going to be doing a few different stains this semester. And one of the things that I mentioned that many of our stains that we're going to be working with are cationic, meaning that they're going to be positively charged. So something like methylene blue or crystal violet, they have a positive charge. They tend to work better on the bacteria because the bacteria has a negative charge on their cell wall. So if you think about opposites attracting, that cationic dye is going to be able to stick to it so that we can see that contrast and we can see those different colors as well as differences between cells. So a simple stain just uses one type of dye. So if we use crystal violet, all of the cells, no matter what their characteristics are, would all be purple. And it's going to give us limited information. It's going to allow us to see the shape and arrangement. A better stain for our time and efforts would really be to do a differential stain. This uses a series of dyes and it allows us to see shape and arrangement, but then it can also allow us to see differences between the cells. So for example, with our gram stain, and the picture that I have in the slide is of a gram stain. So we can still see shape and arrangement, but it's allowing us to see the differences so that we can pick out the gram negative and gram-positive bacteria. Another differential stain is the acid-fast. This would allow us to see if the organism resists the acid wash and is an acid-fast bacteria. We can also do a spore stain to see if the organism makes a spore. Now, some other stains like a negative stain to see capsule or a stain to see if the bacteria has a flagella, these would also be differential stains, but we're not going to be doing those. We're really going to be doing our gram stain, acid fast, and spore stain. Now, coming into micro, we're probably more familiar with eukaryotic cells like plant and animal cells. And at first, we did a little comparison of prokaryotes versus eukaryotes. Prokaryotic cells like bacteria, they are very simple cells. They don't have a nucleus. They don't have fancy membrane-bound organelles. Eukaryotic cells are going to be more complex. We did talk about some similarities. So one of the things that you should be able to do is a little compare and contrast. So write out all of the similarities. Write out all of those differences. Anytime that you're asked to compare and contrast, you will always want to think about similarities. You always want to think about differences. And that's actually where we started with chapter 5. And here I put that little Venn diagram in here so that you can see those similarities and differences. So practice with that, compare and contrast. The other thing that we talked about and we were also able to see in lab were the different shapes and arrangements. Now, the 2 most common shapes that we will see this semester, the coccus, which is the circle, the round shape, and the bacillus, that is that rod shape. We also talked about some different shapes like vibrio, spirochete. Some were actually able to see the spirillum. They looked like these very thin, wavy red lines when we were looking at our slides underneath the microscope. We also saw some different arrangements. So we tend to see more arrangements with cocci because those circles can get stuck at different points on the circle as it's dividing. If you see to the right, our bacillus, we have fewer arrangements, and that's because when they divide, they get stuck end to end. So those are the different arrangements and shapes. If you like to draw, it might be a good idea to draw out the shapes, draw the arrangements so that you can recognize those. One of the things that you're going to have to recognize is that I will show you a picture and you are going to have to tell me the shape, the arrangement, and the gram reaction. So the more practice you have with that, the better. Now, what makes the difference between these cells when we were talking about the differential stains, if it's gram-positive, if it's gram-negative, or if it's an acid-fast, it has to do with that cell wall. So here you can see the differences between our gram-positive, gram-negative, and acid-fast bacteria. Gram-positive bacteria, they have that thick layer of peptidoglycan. The reason that they look purple is because that crystal violet will adhere to that peptidoglycan. Gram-negative bacteria, they will take up that counterstain, safranin or safranin, however it is that you want to say it, and that dye is red, so that's why they will appear red. Down below, you can see our acid-fast cell wall. Now, they have a thin layer of peptidoglycan, but they have that thick layer of mycolic acid. That's that lipid, and that lipid layer acts as sort of protection. So these are tough customers. They are tough to kill, so difficult to treat, also difficult to kill with disinfectants. They are also slow growers. Because of that mycolic acid, it's difficult to get nutrients in and waste products out. So here's a little comparison of the different cell walls, and we would be able to tell that based on those differential stains. So here's a generic picture of a bacteria prokaryotic cell. You should be familiar with the different parts. Again, simplified, not a lot to it. Certainly eukaryotic cells are going to be more elaborate. They're going to be more complex. One of the things that we talked about is that bacteria can have flagella. So we talked about the different configurations of flagella. So I've got a picture there of the different types. So you should be able to recognize those types. Again, if you like to draw things out, that's something that you could draw out and then write the names. And one of the things that we talked about that they do with the flagella is that they can move towards good things like nutrients and away from harmful things. And this process is called chemotaxis. I like to think of a chemical taxi. So that bacteria has receptors. It can sense that substance in the area. And then based on how it moves the flagella, if it's good, it can move towards it. Or if it's bad, it can actually tumble away from it. And I showed you the little video of the bacteria kind of running and tumbling. And it's more like a meander than it really is like a straight run or a straight run away from something. And the way that they can either run or tumble depends on how they move their flagella. So if they move it counterclockwise, that's going to result in a run. So I always remember counterclockwise has an R in it. Run begins with R. Clockwise, that's going to result in a tumble. If you want to go back, feel free to look at that video. You can also look at some different videos on YouTube. I'm just going to warn you not to go down that rabbit hole of chemotaxis because it can get quite complicated. Now, I mentioned that our eukaryotic cells are very, very complex. And so we talked about this theory of endosymbiosis, that chloroplast and mitochondria, those organelles, actually evolved from smaller prokaryotic cells that were gobbled up by those larger prokaryotic cells. We also looked at some of the different evidence. So we should know that there definitely is evidence for this theory with our chloroplast and mitochondria. That was actually one of your assignment questions, that you had to list at least 3 of those. So you want to be familiar with this theory. We also talked about cells being at the mercy of their environment. Our cells, bacteria cells, in order for a cell to be successful, it has to have a high surface area to volume ratio. And I put this little picture of cubes just so that you can see the bigger the cell gets, its surface area to volume ratio starts to decrease. So our prokaryotic cells are very small, so they actually can maintain that high surface area to volume ratios. Our eukaryotic cells, because they're bigger, they have to employ some other things to help increase that surface area to volume ratio. So one of the things is flattening, folding. Anytime that you fold things in biology, you know we increase the surface area, and compartmentalizing. So by the eukaryotic cell having those organelles, that's going to increase its surface area to volume ratio. Chapter 7, we got into the growth of bacteria and how we can actually measure that growth. So bacteria replicate very quickly, very simply, and that process is binary fission. So they are going to clone their DNA and split into two. So those two daughter cells that you see at the bottom of the picture, they're identical to each other, identical to the parent cell. So not a lot of genetic diversity. Because this is a simple process, they can reproduce very quickly. We also talked about the growth curve. And from the growth curve, we could actually calculate generation time. Generation time is the amount of time that it takes for a bacteria to double its population. So because these bacteria replicate by binary fission and it's very simple, their generation time can be anywhere from like 20 minutes to 2 hours. One of the things that you should be able to know and label and recognize, be able to describe the different phases. So again, this is another thing that you can draw out. So we talked about lag phase. This is also called the period of adjustment. Bacteria is getting used to the environment. Log phase is when that cell is dividing, and so now we're increasing that population exponentially. Stationary phase, as resources start to run out, the number of cells dividing will equal the number dying off. And then death or decline phase, now they're dying at an exponential rate because they've run out of resources. So be able to discuss the different phases as well as label them on a graph. This is something that you can draw out and you can actually practice. One of the things that I mentioned was that if we were growing an endospore former, we would probably start to see some of those endospores in stationary phase, even more so as we got into death or decline phase. So how could we actually measure bacteria? So before we could actually plate out our bacteria, because we have no idea how much bacteria we have, we would have to do a serial dilution. And this is going to get us a range of countable bacteria so that we get in that sweet spot of 25 to 250 CFUs. This is something that we're going to be doing this semester in a lab. So we want to make sure that we cover all of our bases, which is why we would do several dilutions. Then once we did our dilutions, then we would plate it out. Now there's a couple of different methods for plating out our dilutions. We could spread the bacteria on an already formed agar plate, or we could add our dilutions and then pour the media on top of it. That is actually a better method, the pour plate method, because it's going to allow us to capture both the aerobic and anaerobic bacteria. And that's actually the technique that we're going to be doing. It's also going to give us the opportunity to pour plates. Now, I did mention a calculation in class. I am not going to ask you about the calculation on the exam. That's going to be coming when we get to that lab. I also mentioned being able to measure the turbidity or cloudiness with a machine called a spectrophotometer. And this is actually what I use to measure the growth of the bacteria that I characterized. So I used a plate with about 100 wells in it so that I could get a lot of data on a lot of conditions that I put the bacteria in. Now, all bacteria are going to have their optimal conditions. All bacteria are going to need water. They're all going to want to have a certain temperature, oxygen level, also pH, just like we have our optimal environment. So we started talking about some of those physical factors. We talked about pH. So organisms that like it a little more acidic, they would be acidophiles. Alkalophiles like it more basic. Neutrophiles, they're going to like it somewhere in the middle. So around neutral. Now, most bacteria that would be pathogenic to us would be neutrophiles because in most places of the body, we have a neutral pH. We also talked about temperature. So depending on the temperature, psychrophiles like it a little cooler. Mesophiles, again, would be more pathogenic to us because we're mesophiles. So they would need to be able to tolerate our body temperature of 37 degrees Celsius. Some are more extremophiles where they would like it really hot. Most bacteria that we're going to be working this semester, they either like it at room temperature or at body temperature. Another thing that we're going to be testing for are oxygen requirements. So we're going to be using a special media, a broth that has an oxygen gradient. So there's a lot of oxygen at the surface and no oxygen on the bottom. Depending on where we saw cloudiness, that would tell us the organism's oxygen requirements. If we saw growth at the top, that would be an obligate aerobe. It has to have oxygen. If we saw growth at the bottom, that would be an obligate anaerobe. Some that are very flexible, they're very facultative, we would see growth throughout the entire tube. Now, we know that bacteria are going to be at the mercy of their environment, and they're going to have certain conditions just like our cells. So we did talk a little bit about tonicity. Bacteria are going to like an isotonic situation, just like our cells. One of the things that I mentioned is that before refrigeration, they actually took this theory and put it into practice to help with food preservation. So if you think about food that is in like brining or curing, or maybe something in a high sugar concentration, that is going to help to slow down bacterial growth, because when you put that bacteria in a hypertonic or hyperosmotic situation, that water is going to leave the cell. It's going to go from high to low. Water leaves the bacteria, it's not going to be able to survive. So this is something that we've been able to take advantage of with food preservation long before refrigeration. I mentioned our acid-fast bacteria. We also talked about our endospore-forming bacteria. There you can see a picture of our endospores and our vegetative cells. Endospores are dormant, so they have got the memo that times are tough. So low water, no nutrients, and they're going to go into this dormant state. Vegetative cells, those are the pink cells. They're growing and functioning normally. I also have some examples. We talked about a couple of examples like Clostridium botulinum. We also talked about Clostridium tetani, and I also mentioned C. diff. Which is certainly a concern in healthcare. The other thing that we talked about was growing bacteria and the different types of media that we can use. A selective media is going to select for a particular organism and inhibit the growth of other types. A differential media is going to allow growth of bacteria, but it's going to allow us to see differences. Sometimes we can actually have both, where we're using a selective and differential media. If you take a look at the picture to the right, you can see the growth on different agars. So if we were to grow Staph aureus and E. coli on a nutrient agar, they look very similar. If we were to grow them on MacConkey, you can see that that's going to select for the gram-negative E. coli. If we were to grow E. coli along with another gram-negative bacteria, you can actually see the differences. So you can see where E. coli is kind of pink, where that Salmonella is kind of yellow. So sometimes it's actually better to use a selective and differential media because we can cover more bases so that we can see those differences between organisms. The last chapter we talked about classification and taxonomy, and I gave you the order of classification. So the big idea with this is that there's that upside down triangle. If we look at the big part of the triangle, organisms in that group are going to have some things in common. As we get closer to the triangle, they're going to have more and more things in common. So the order of classification: kingdom, phylum, class, order, family, genus, species. You can see there's another mnemonic here. The mnemonic that I learned years ago in 7th grade was King Philip came over for good spaghetti. This mnemonic is King Philip can only find green socks. I still say this mnemonic in my head, so whatever works for you. Just know that the big idea is that at the top where that triangle is widest, they have some things in common. As we get closer, they have more things in common. So looking at that upside down triangle, organisms in the same family are going to have more things in common than organisms in the same class because they're closer to that triangle. The other thing that we talked about is this binomial nomenclature. So this two-name system, and that two-name system is genus and species. We need to know how to represent that. It needs to be italicized. If you can't italicize it, then you need to underline it. Genus is always capitalized and species is always lowercase. We call that the Latin or the scientific name. The last thing, we talked about many ways that if, let's say that we found a new organism, we could find out if it's a brand new organism or if it's related to another organism. And these last two techniques really help us to determine whether or not we have a new organism or the identity of that organism. And these are two techniques that you should be able to talk about. So you definitely want to practice writing this out, saying it. I always say that if you can explain it, then you know it. And I have these two pictures. So the one is on DNA hybridization. So you can see where there's DNA with different colors. One is red, one is green. So these two DNA strands were taken from different organisms. If you can put them together and it's a perfect match, like you see on the right side, all of those sequences are matching up. It's the same organism. If it fits, but there's still a couple of spots that don't quite make it, they're not a perfect match, then we know it's somewhat related. If it's a hot mess and nothing is matching up, we know they're not related at all. We can also do this with proteins in gel electrophoresis. As we separate out the proteins, they will separate out based on size. If you see on that bottom picture, there's little lines, and that is giving us the protein profile. If protein profiles match up perfectly with two organisms, we know that they're the same. So again, by looking at that similar sequence of DNA and proteins, this can also tell us how related organisms are. So what you want to do is you want to make sure to practice writing these out. If you like to draw things out. I always say that if you can explain it, then you know it. So practice, practice, practice. I hope that this PowerPoint offers you some different pictures, different ways of organizing the information that helps you to study for the exam.